Welcome you all in a new lecture series, and here we are going to discuss metabolic fate of ammonia. So as we know that the ammonia which is produced in a various tissue, and here it is commonly produced in a brain, kidney and muscle, and this ammonia it is transported in a circulation and it goes to liver, where in a urea cycle you will get a urea. This is the structure of urea. So this metabolic fate of ammonia, it will be discussed under the following heading. So first one is the production of ammonia, how the ammonia is produced. The next one is the transport of ammonia, how the ammonia is transported. Next step is a conversion of ammonia to urea, and at the end how there is a disposal of urea. So first of all we will discuss production of ammonia. Now here you can see that there are the processes like transamination and deamination. We have seen that ammonia it is produced by through the process called transamination and deamination. Here there is a in transamination there is only a transfer of amino group. Here the alanine is there, and this alanine it is from protein. The source may be endogenous or exogenous. So this alanine it is produced by the process of transamination. That amino group it is transported to alpha ketoglutarate, and here you get the glutamate. This glutamate now it participate in a deamination reaction, where by the action of DDH we get ammonia through alpha aminoglutarate and alpha ketoglutarate. Ultimately at the end you get the ammonia. Similarly, by the process of oxidative deamination, by the action of enzyme L amino acid oxidase, D amino acid oxidase on this L and D amino acid respectively, here you will get ammonia. And again, we have another pathway, that is a non-oxidative deamination, where you will get ammonia. So here you can see that this is a conversion of glutamate to ammonia. ये जो pathway है, this pathway is a major pathway. This one is a major pathway, and other pathways are minor pathway. And in this series, we have another pathway, that a small amount of urea it is produced in a intestine by the action of intestinal bacteria on a urea. So by the action of urease enzyme, there is a formation of ammonia, and this ammonia, which is a small quantity, it is absorbed from intestine, and here it contribute to the formation of ammonia. So here you can see that these are the pathways by which you are getting the ammonia. Now these are the steps involved in formation of ammonia. Then now, here you can see that by the process of transamination and deamination, here you will get a ammonia. This ammonia, it never exists in a form of ammonia form. It get converted into the ammonium form. And this ammonium form, it combines with the glutamate so as to form a glutamine. And the enzyme is glutamine synthase, and this glutamine it goes into the liver, where by the action of enzyme glutaminase you will get glutamate and ammonium ion. Okay, then what is the metabolic fate of ammonia? Now we have ammonia it is entering into the liver, and what happens with ammonia, let's see. So this ammonia it will go in a urea cycle. So the ammonia which is produced in the most of the tissue, and it exists in a ammonium form, it immediately combines with the glutamate, it gives glutamine, and also this alanine, it carries amino group. So this glutamate, glutamine and alanine, ultimately they are responsible for production of ammonia, and that ammonia it is transferred to liver, where it participates in urea cycle. So the ultimate conversion of ammonia to urea is takes place in a liver. And now how the transport system, now wherever the ammonia which is produced it has to transport. So there are the two mechanisms which are explained. The two mechanisms are available in a human for the transport of ammonia from peripheral tissue to the liver for its ultimate conversion to urea. So in this series we have First one is the transport of ammonia in a form of glutamine, which we have already seen, but this pathway is very important as it is produced in a kidney and brain. As we know that ammonia is a highly toxic, so it ultimately converts into glutamine. This glutamine, it is formed by the enzymatic activity that ammonia enzymatically combines with the glutamate so as to form glutamine, and the enzyme is glutamine synthetase. So here you can see that there is utilization of 1 ATP, and here there is a formation of glutamine, and this glutamine it goes into the liver where by the action of enzyme glutaminase you will get glutamate and ammonium ion. Okay, so this is the first pathway, first mechanism. Now the another mechanism is transport of ammonia in a form of alanine. So here you can see that pyruvate along with this ammonium ion by the process of transamination, here you will get alanine, and this alanine it is transported from muscle to the liver, and the cycle is called as glucose alanine cycle, and here there is a release of this ammonium ion in the liver which is utilized for the urea cycle. So this is glucose-alanine cycle. It will be discussed in a big detail in a carbohydrate metabolism. Then the urea it is the end product of protein metabolism. Now the our important step that is the formation of urea. So this urea cycle, the site of urea formation is a liver, and it is produced exclusively in a liver, and the site of excretion is our kidney, and some part of the urea it is excreted through your intestine. Okay, so what are the characteristic feature of this urea cycle? Where it involves a fire reaction which includes ornithine, citrulline, arginine, and aspartic acid. So let's see how this urea cycle takes place in a liver. Now here you can see that this is an important step in a formation of urea cycle. So the urea which is an end product of protein metabolism. So here this is the structure of urea, and at the end you will get the urea, and what are the precursors here? So here this is the ammonia which is in a ammonium ion form. This is a bicarbonate. It is from carbon dioxide, and this carbon dioxide it is released by the Krebs cycle through oxidative decarboxylation. And here this is another amino group. It is from L-aspartate. Now, what are the enzymes required for urea cycle? So here this is your hepatocyte where you have a mitochondria, and this is a cytoplasm. In mitochondria, we have two enzymes. One is the carboyl phosphate synthetase 1 and ornithine transcarbamylase, where this enzyme carboyl phosphate synthetase 1 which is required for the synthesis of carboyl phosphate, and second enzyme that is the ornithine transcarbamylase which is required for the synthesis of citrulline. In cytoplasm where we have a enzyme argininosuccinate synthetase which is required for the formation of arginosuccinate. The second enzyme is the arginosuccinase which is required for cleaving this arginosuccinate, and the last enzyme that is the arginase which is required for cleaving arginine to form ornithine and urea. So here you can see that this is your mitochondria and this is the cytoplasm, and here it takes place, these two steps takes place in a cytoplasm as well as in a mitochondria of hepatic cells. So first step is our, this is the urea cycle, this is your mitochondria. Now here this is your mitochondria in which here there is an entry of carbon dioxide and ammonium ion. So this ammonium ion it is from your glutamate by the action of enzyme glutamate dehydrogenase, where there is a formation of alpha ketoglutarate and the ammonium ion, and this ammonium ion enters into this pathway, this cycle, and here by the action of enzyme glutaminase we get glutamate plus our ammonium ion. Now here this carbonate and this ammonium ion, they condense together. Here you can see that the first group of urea, it is derived from the free ammonium ion in urea cycle in the mitochondrial matrix, and this ammonium ion, it combines with the carbon dioxide to form carbamyl phosphate. This is the first step, and this step, it is catalyzed by the enzyme carbamyl phosphate synthetase 1, and it is driven by hydrolysis of ATP to ADP, where one ATP, it goes to the phosphate, and another ATP, it is used for the this energy, where which is utilized for the condensation of this carbon dioxide and ammonium ion so as to produce carbamyl phosphate. And we have seen that the source of this carbonate, it is a carbon dioxide. It is from Krebs cycle, which is produced by the oxidative decarboxylation, and the ammonia, it is from the amino acid by transamination and deamination, which is used. So here ultimately you have the carbamyl phosphate. Now here we can see that we have a carbamyl phosphate, and here the activity of this enzyme carbamyl phosphate synthetase 1, which is regulated by the N-acetylglutamate. This N-acetylglutamate stimulates the activity of carbamyl phosphate synthetase 1, and here you get a carbamyl phosphate. And this carbamyl phosphate synthetase 1, it is a regulatory enzyme of urea cycle. Now we have a ornithine. This ornithine is same as that of our oxaloacetate in a Krebs cycle. Now this ornithine, it is here, it is transferred from cytoplasm to mitochondria. Here this ornithine is transferred from cytoplasm to mitochondria, and here it combines with carbamyl phosphate. Here it combines with the carbamyl phosphate to form L-citrulline by the action of enzyme ornithine transcarbamylase. Here this ornithine transcarbamylase, it is a mitochondrial enzyme which transfer the carbamyl group from carbamyl phosphate to the ornithine, and here you will get a L-citrulline, and during this step there is the release of phosphate. The phosphate, it is a release in this reaction, and here we will get a L-citrulline. Okay, now this L-citrulline, it is formed, and this cycle also called as a ornithine cycle. This cycle, the urea cycle, is also called as a ornithine cycle. Okay, now next step is a synthesis of argininosuccinate. Here we want argininosuccinate, and the enzyme is argininosuccinate synthetase. The enzyme is a argininosuccinate synthetase, and here we are using ATP. So here you can see that the L-citrulline, which contains with the L-aspartate to form argininosuccinate, so as to form argininosuccinate, and this is a condensation reaction. For this reaction, the energy is driven by the hydrolysis of ATP. This ATP, it is hydrolyzed to AMP and to inorganic phosphate. So here this this is a pyrophosphate. Here you can see that this is a pyrophosphate, which ensures that this step is irreversible. This condensation of this L-citrulline with the L-aspartate, it is irreversible, and here there is a formation of our argininosuccinate. Okay, then next step is a cleavage of this argininosuccinate. The argininosuccinate, it is cleaved by enzyme argininosuccinate lyase, or it is also called as a argininosuccinase, to yield L-arginine and fumarate. Here there is a formation of L-arginine and fumarate, where this fumarate, it enters into the Krebs cycle. This fumarate, which is formed here, which enters in our Krebs cycle or TCA cycle, as it is a intermediates in a TCA cycle, and here you will get this arginine. Now, next step or last step is a cleavage of L-arginine. So here this L-arginine, by the action of enzyme arginase, you will get urea. Here there is a release of urea, and this urea, which is our end product of our protein catabolism. Okay. Now here you can see we have discussed that this arginine, it is a semi-essential amino acid. Why? Because a small amount of arginine, it is a produced in our body. But in the liver we have an enzyme arginase, so there will be no arginine, it will form in a liver. But in case of our kidney, the same cycle takes place, but there is a formation of the arginine, but the enzyme which is required for cleaving this arginine, the arginase, which is absent here. That's why a small amount of L-arginine, it is synthesized in a kidney, and here that's why it is called as a semi-essential amino acid. Now, whatever the urea which is formed here, this urea, it will excreted in a urine and a feces. By kidney, it goes to urine, and some part of urea, it comes into the intestine, and here it is a release in a form in a feces. Okay, this is about a urea cycle, and ultimately here you will get a ornithine, and this ornithine again enter into this cycle. Okay. Now, here you can see that this is our urea cycle, and this is the Krebs cycle. So the intermediate of this Krebs cycle, that is the oxaloacetate, it get converted into aspartate, and this aspartate combines with the citrulline by the process of condensation, and here you will get a argininosuccinate. Now, by the action of enzyme argininosuccinase, you will get a fumarate, and this fumarate, it enters into the Krebs cycle, where it participates in the Krebs cycle, and here you can see that this cycle, it is just like a bicycle. That's why it is called as a Krebs bicycle. Okay. So the urea which is formed here, it is excreted in the urine, and the human being excrete about 10 kilogram of urea per year. Now, what is the energetics of urea cycle? So here you can see that the 4 ATPs are consumed in the synthesis of each molecule of urea. So here the 2 ATPs are needed to make carbamyl phosphate, where the 1 ATP serves as a source of phosphate for the carbamyl phosphate, and second ATP it get converted into ADP plus inorganic phosphate. This ATP which is used for the condensation reaction. Then 1 ATP is required to make argininosuccinate, where the 1 ATP it is hydrolyzed into the AMP and two inorganic phosphates. So the cleavage of this inorganic phosphate is also called as a pyrophosphate, which ensures the irreversibility of this condensation reaction. So here this 1 ATP is equivalent to 2 ATP. So the total ATPs are utilized is a 4 ATP. Okay. Now here in your exam you have to write this, that ammonium ion combines with a carbon dioxide, plus there is the entry of L-aspartate, plus 3 ATPs are utilized so as to produce urea, and here there is a release of fumarate with there is a 2 ATP ADP plus 4 inorganic phosphate plus AMP. So at the end, after a calculation, it will be like this. So you have to write this in your energetics of urea cycle. Then what is the regulation? How the regulation of urea cycle takes place? So here we have a carbamyl phosphate synthetase 1, which is a allosterically regulatory enzyme of urea cycle, and which is allosterically activated by N-acetylglutamate. This N-acetylglutamate, it is a allosterically activate this enzyme. So what is this NAG? It is synthesized form acetyl-CoA and glutamate by the action of enzyme NAG synthase, and which ultimately activate CPS, that is the carbonyl phosphate synthetase 1. It has only this action, it has no other action. Now the synthesis of this NAG, it is increased by whenever we take in your whenever the intake of protein is more. If you are eating more protein, that protein which contains arginine. This arginine, it activates the synthesis of this N-acetyl glutamate synthase. This arginine, it activates the synthesis of NAG synthase, and this NAG synthase, it will activate carbamyl phosphate synthetase 1. And during starvation, again there is a activation of this NAG synthase so as to produce NAG and to activate carbamyl phosphate synthetase 1. Now here you can see as there is the entry of protein, that protein responsible for increasing your glutamate level. Again the glutamate is also elevated, and here there is increase in synthesis of your N-acetyl glutamate, and this N-acetyl glutamate allosterically activate carbamyl phosphate synthetase 1. This is the another mechanism. So this is all about urea cycle. We will discuss urea cycle disorder in our next lecture. So please like, share and subscribe. Thank you.